put. Anyway, so Dr. Jon Chamberlain is a senior lecturer and researcher in collective intelligence, information processing and marine technology in the School of Computer Science and Electronic Engineering. And he's also the director of the Marine Technology Research Unit and collaborates a lot with life sciences. And I'm really excited to hear all about all the work you're doing and your students. And so, yeah, take it away, Jon. Thank you for being here.
Yeah, just because we've got computer scientists in the room doesn't mean that we can actually get the Zoom room functionality working properly. Yeah, so thanks for the invite to come over here. It's great to talk to you. I know quite a few of you already from various different projects and things we've worked on. And really, I thought it'd be a great opportunity to kind of give you an insight into different types of projects that we've worked on in collaboration within life sciences, also with some of our NGO partners that are very much sort of aligned to the kind of things that we do in life sciences. In a desperate attempt to be sort of interdisciplinary and non-siloed. So it's a bit of a whistle-stop tour, really, of everything we do. And you know, if anything sort of piques your interest, then do get in contact afterwards. I have with me my PhD student, Mark Roper, and also my master student, Rachel Lowe, who are going to present some of their work as well. And I'm also be presenting some work from Jess as well, Jess Ryan. So it's no surprise to most of you here that marine systems are under threat and have been for a while. In particular, I'm quite interested in the tropical hard corals that are very slow growing. And if you look in this that first picture, you can see the kind of ecosystems that these branching corals are supporting. This high level of complexity provides a habitat and a home for these orange amphias and for the feather stars to pop out of the into the current. You can see all the things down the bottom that are growing with sponges and that kind of stuff. So it's an example of a healthy ecosystem. And when that starts to degrade and bleach and then eventually erode away, you lose that level of complexity, you lose that level of diversity on the reef. That's kind of a well-known aspect. So one of the things that we're very interested in is whether artificial intelligence and technology in a broader sense can actually help us monitor and try and address this threat. First, I'd like to just kind of talk a little bit about what is artificial intelligence. It's the only kind of ticky slide really in there, but it's a term that's bandied around a lot, and I think it's kind of overused really. In its strictest sense, artificial intelligence is relates to intelligent behavior of agents, whether they be human or animal or computational. So it really combines things like computational psychology, philosophy, things like that. But really we use it in a broader sense to think about things like machine intelligence. When you look at the ACM definition of artificial intelligence, it includes subdisciplines such as automatic programming, expert systems, theorem proving, machine learning, natural language processing, robotics and embedded systems, computer vision and scene understanding, and distributed and collective intelligence. And these are all aspects that we work on within the MTRU, which is why I talk about artificial intelligence in that kind of broader, less specific sense. And it all kind of kicks off really, certainly in my mind, in computer science, from some work that happened about 5-10 years ago. Some colleagues built some robotic fish. So these are autonomous robotic fish swimming around in the London Aquarium. And they can do sort of object detection and can kind of avoid things, but just generally they look quite nice and they swim around. And it was part of a project to detect marine pollution in ports, so that they actually went a bit further and actually made their robots look a lot like fish. And the idea was that was a collaboration between life sciences or biological sciences back then and engineering, and that's really what we want to try and do here. So our current research looks at two main areas. First is remote sensing and monitoring, and that incorporates things like photogrammetry, rapid surveying, measuring of 3D models, that kind of stuff. And then the second big area is scientific engagement and citizen science. So things like interactive virtual environments and 3D printed models, those kind of things. We're always looking for different ways to collaborate and different ways to innovate. So, Ken, so if anything piques your interest, please let me know. We're going to talk quite a bit about 3D models. So I just thought I'd briefly mention some terminology related to the models. So you can see on the left of these images of rabbits, is you have a rabbit made from points made in 3D space, and that's what's called a point cloud. So individual points with X, Y, Z coordinates gives you the illusion of the outside of a rabbit. You join those dots together, you create polygons, and the polygons all together is what's called a mesh, and that's what you see in the middle. And then if you color those polygons in and smooth them out of it, that's where you get your photorealistic 3D model, which is what you see on the right. So that's called a textured model. In order to get those individual points in 3D space, you can use something called 3D photogrammetry. It's not the only method you can use, but it's the one that we use quite a lot in our research. It looks quite a technical diagram, but just kind of ignore all these sort of algebra stuff. There are 3 viewpoints here, and they're called cameras, rather confusingly. But imagine those are images, 3 images taken of that cube. And what the photogrammetry software process does is it tries to identify the features of that cube and locate them in each of those images, and then it can triangulate where those points are within 3D space, roughly how it works. There are 2 approaches that you can use with photogrammetry. The structure from motion, which builds a detailed 3D model, and that's mostly what we use in our research. You can also use visual SLAM, and that's the kind of thing that a self-driving car would use. So it builds the model in real time and locates you within it. But we don't use so much of that, so much of visual SLAM. Photogrammetry is actually commonly used in surveying, certainly for those that have been out to Indonesia and Croatia out on field trips with us. It's a standard method for Operation Wallacea, for the monitoring that we do. And a common way to do it is to lay out a 2-meter square quadrat and then move a GoPro camera across that in a systematic way to collect images. And put it through a bit of software and it produces a 3D model. And what you can see there is actually a 3D model. That's not an image, or it is an image. It's an image of the 3D model. These blue rectangles are each viewpoint, each camera, as they call them, of every image that was taken of that scene, and they're joined together to produce the 3D model. You can use one GoPro, but why use one GoPro when you can use 20? So this is one of the first bits of research that we did in 2017, was to stick 20 GoPros on a plastic dodecahedron frame. And that allows us to collect imagery from lots of different directions at once. It's called 4-pi steradian, if you want to take the term. And it actually is very effective at collecting data for building a 3D model. This is a 90-meter wide hammerhead jetty, it's the underside. So the legs that you can see here are the legs of the jetty. And we just ran that ball of cameras through there once to collect the imagery. And it's enough to build the seabed, which you can see quite clearly, and the pilings of the jetty as well. The purpose of that research was to show that using multiple cameras, you can speed up the surveying process. Really, that was what we were trying to show. And so compared to doing a 2-meter squared process in the usual way, which would take around about 5 minutes, if you just use 5 cameras and just go over it once, you get the same model, actually the model is slightly better, but it takes 45 seconds. So it's considerably faster than what you do. So really that was the purpose of making the multi-camera model. Once you have your 3-dimensional model, now you can do some really interesting stuff. And this is the kind of the interesting part of the talk, really. So one of the things you can look at, because you have your 3-dimensional model, is you can look at the complexity of the shapes that are within the area that you just surveyed. You can accurately measure the volume, not just of the whole scene, but of individual parts. So individual coral bommies or that kind of thing, you can measure their volume quite accurately. You can also measure color, which is very important if you're trying to measure coral bleaching or disease, and also to measure the different types of substrate cover and different percentages of substrate. This is just one example of how you would measure complexity computationally. So normally, if you're doing this in situ, you would use a tape and chain. So you'd use a piece of chain of a known length, probably 2 meters, you drape it over the reef, and then you measure how far it's gone. And then you just do a ratio of however long it is, versus however far it got. And that gives you your rugosity measure. But of course, you're having to drape that over the delicate corals and delicate soft corals, causing damage to the reef by taking measurements. And it's quite hard to get it into nooks and crannies and underhangs and that kind of stuff. So if you have a three-dimensional model, you can actually slice that model up computationally and actually measure the distance very, very accurately. And that's what you see in the bottom right-hand corner of this image here, is slices through the reef, basically to create those rugosity measurements. So another case study about how this works in the field was some work that we did with Natural England in 2018. Some divers reported that the fishing activities on the chalk reef off North Norfolk was causing the chalk to be damaged. And the chalk reef has been designated as a marine conservation zone. So Natural England were concerned that, they had an obligation to do something about it. So we got involved to actually see if we could help them gather more evidence about the impacts of human activities. So we went out and we surveyed the chalk reef with them. This is a double chamber parlor pots that is very commonly used in Norfolk. They're different around the country in different ways of fishing, but this tends to be what they use. And they're quite heavy because there's quite a lot of wave action. They're deliberately heavy to sit on the seabed. But of course, because they're heavy, it means they're scraping along the chalk, and you can see underneath that pot there that that's been moving around a bit and it's scraped off the epitherm. So we did surveying with them, and we only had 4 dives, but within the 4 dives, we actually managed to collect enough data to produce a meaningful report. We managed to categorize 11 different types of damage, some more severe than others, some more prevalent on the reef than others as well. And we did a similar thing to what I mentioned about measuring rugosity. So we took those 3-dimensional models that we took from the chalk reef, we sliced them up, and we got some numbers that we could then correlate with the types of damages that we saw. And we did actually find that there was correlations between damage and the rugosity and complexity of the reef. Part of Jess Wright's PhD work was to look at different ways of measuring that type of complexity. So on temperate rocky reefs, you have a different type of shape of reef than tropical coral reefs. If you can imagine a tropical coral reef, it tends to be complex in all different directions, whereas rocky reefs tend to have gullies and stacks and arches, these kind of features that kind of tend to be quite linear. And they were not being captured adequately with the kind of complexity metrics that we're using. So Jess came up with this idea of using a computational technique of measuring relief within a bounded area. Basically, you take a square and then measure the highest and lowest points in the inverse. But that's a work in progress. I think more importantly, our work had a big impact on the fishery in North Norfolk, and unfortunately it wasn't a positive one because we demonstrated that fishing activity was causing damage, and therefore action has been taken with the possibility of threatening to close down the fishery. We didn't want to happen, but it has instigated considerable activities from the fishing authorities and Natural England as follow-up actions to try and minimise chalk damage. Both in the short term, but also for the sustainability of the reef. Once you have a 3-dimensional model, you're quite interested in being able to identify the individual parts of that model and what you're seeing there. And this is what's called semantic labelling. So here you can see 2 images. The image on the left is just a standard 2-dimensional image. The image on the right has polygons drawn on there to indicate that these are certain types of things. So the yellow is, looks like soft coral, and the red looks like branching coral. Okay. So that's what we mean by semantic labeling. We're giving meaning to parts of the image. Now, to do this on 2-dimensional images is relatively straightforward for computers to do, and straightforward but boring for humans to do as well. So really, we want the computers to do this quite reliably, but actually for marine images, it's very difficult. For identifying cars and faces and that kind of stuff, it's pretty you get good results. But for marine images, you can imagine that the morphology of the type of species that you see is very varied. And as such, it's a very difficult task to do, which means it's a good task to invite research teams to work on. So we provide data sets for international research teams through a competition called ImageCLEFcoral, been running for 4 years. They get a training set and they develop algorithms using different approaches to try and solve this problem. And then we give them a test set and we give them the results based on their algorithms. And to cut a long story short, it's a very difficult task, and no one's been able to produce anything that is, I would say, good enough to be used as either a commercial product or a research product. You wouldn't want to rely on data. There's too much error, too much variability. The MSc Tropical Marine Biology students in life sciences helped us create that data set over the last few years, pre-pandemic and during the pandemic as well. And that is actually just doing the training of the students as well. So it's a good opportunity to get involved, to learn about marine species ID in different places around the world, to learn about different methods of doing those annotations. Those annotations are then checked by validators. And we also have automatic software checking as well to make sure that we have the best possible data that we can. We have a new project starting with Philippe as well, using Lightsheet Microscopy, and basically using AI techniques to try and improve on the imagery that he's collecting through Lightsheet Microscopy. It's just started, so we don't have a lot of information as to and results. It's a really exciting project. We're working with Philippe on that one. So those are some ways that we can use photogrammetry to build three-dimensional models. We can semantically annotate them. Now we also have a model. We can also track things in 3 dimensions. And this is the work that my new PhD student, Mark, is working on at the moment. So I'll hand over to him. We'll talk about that.
Hi, I'm Mark. I'm a first-year PhD student. I'm working on 3D underwater tracking. So the basics of this is how can we get real-life data, tracking data, out of video imagery? So to start with the basics, we have 3D tracking is the ability to track an item in XYZ coordinate space. So in order to be able to do this, we need to have 2 cameras or more. There's 2 ways of doing this. You can either have a binocular stereo vision, like our eyes, we have the 2 cameras next to each other, or as we can see on the screen, we have 2 cameras which are distal to each other. This allows a slightly greater amount of space and viewing angle between the objects. But in effect, any object which appears in both camera lenses, we can 3D track, as long as we have calibration and synchronization between the cameras. So the first question is, why do we want to do 3D tracking? Most of the research would be done with just a video camera, like we saw earlier. We take a scan of the environment, and if we're looking to say how fish interact with each other, or with prey, or with food sources, we just take a video image and annotate it. The issue with this is we do not get the speed and distance information correctly. So obviously, the closer the fish is to the camera lens, the speed is different to when it's further away, because we're recording speed in pixels, not in actual metrics. So by adding the second camera and doing a 3D space, we can actually get the actual relative velocity and direction of the fish or whatever we're studying as it swims around. The second one is we get a slightly better viewpoint. If we're using distal cameras, rather than having a single view of that item or animal, we can actually see its side and its top view. So again, we might be able to pick out more behavior differences than if we were just taking a single image. The final sort of advantage is with 3D tracking software, you typically do 2D tracking on each of the images that you captured, and then you combine them to create the 3D track. However, with 2D tracking, we have the option, the problem of occlusion. So say the fish swims behind a coral, or you have 2 fish cross each other. Now we have the question, how do we know which path is actually which fish? So with the 3D tracking, we get slightly more information. So we can say, okay, the relative velocity before the occlusion was this speed at this velocity and this direction. After, we've got the new thing. So we've got 2 views, which allows us to get a better view of where the fish was and which tracking trace is which. So that brings us to the next question. Why are we doing high-speed 3D tracking? So by high speed here, I mean anywhere from 120 to 240 frames per second. So we're not into the thousands of frames per second. But with high-speed tracking, we are able to see things which you cannot see with the naked eye and things which you might miss. So in my previous PhD, we actually had, we would doing 3D tracking of honeybees. And what we actually found is they fly very fast, but their antenna move incredibly fast to determine whether or not a food source is rewarding or not. Now, when this was recorded at 30 and 60 frames per second, you cannot see the antenna doing the inspection. But recording it at 240 frames per second, you can actually see the antenna move slowly across, touch the feeder, then reject or accept it, and then the bee lands. Similarly, if you're looking for something in marine ecology, so say we're looking at how do the anemones interact with each other or themselves and other items around them. Now, if you're again recording at 330 frames per second, you may not actually be able to see any of the detail. You won't be able to see the contacts instant and whether it repels or rejects it. So recording at this high frame rate allows you to see far, far more than you would at a slower frame rate. The downsides are you need a lot more light. So obviously your shutter speed of the camera is going that much faster. You need more light in the environment. So the advantage is you get motion blur is removed because you're doing this faster shutter speed. The other issue we have is a vast quantity of data. So we're now recording 8 times as much video footage as we were before. We're having to do 3D tracking on that big data footage, so we end up with a much, much bigger data set. So the question then is, how can we actually analyze this information? So this basically brings together all of the things we've heard about today, and from Rachel's talking a bit, is what we're going to do is take the 3D scans taken through photogrammetry. We're going to combine this with the, so if you look at the bottom left, you have the 3D trap, where in this case, this is actually a fruit fly going around an arena, but this would be equivalent to a fish being tracked through its environment. We can merge this with the photogrammetry and 3D models of the anemones, fish, or whatever else we need to be able to simulate the world. So what this gives us is a recording of what happened. So we can go through and track it again and again. The difference is with a 3D model inside a game engine, we can actually rotate the camera. So now we can see what happened from a completely different view to what was actually recorded. So if we wanted to see what the interactions behind the fish were, we know what the 3D model of the arena is, or the environment. So we'll be able to see how the corals affected what movements the fish made. So by adding all this, we can see a lot more data than purely what's available from the video recordings. So what this allows us to do is to be able to do, like John was speaking about earlier, is AI inference of what the behaviors are. So rather than trying to scan through and record, okay, this velocity and that velocity in this direction, what we can actually say is to an AI, go through all of this footage and all of these 3D tracks and see if you can pull out things which occur regularly. So do the fish always have a particular behavior where they slow down and change direction, or they speed up? And then can you then coordinate that back to, was there another fish in the environment? So is this a escape response or a prey detection response? Most of this is just simply not available with sitting there and watching video footage. So the highlights of my PhD projects. The first one is we're going to be using high-speed cameras. So in order to get proper 3D tracking, we need to be able to calibrate these cameras together, and most importantly, to synchronize them. So we need the footage and the frames to be at the same rates. Now, these could be right next to each other, the cameras on a ROV, which makes it slightly easier. Or you might have 2 cameras floating around in different areas which need to be synchronized. So part of my project is how do we synchronize these 2 cameras together? The second issue for marine environments is it's underwater and it's going up and down. If you're doing any work on dry land, you put the camera on a tripod, it's there, it's stuck, no problem. When we come to do it in marine environments, the ROV, or even if it's on a tripod on the sea floor, it's still being moved around by the water volume. So every time the camera moves, we need to recalibrate and work out how to do the 3D tracking. So part of my project is to use the photogrammetry scan of the reef in order to work out where the camera is for each frame of footage. This then allows us to then recalibrate where the cameras are and reproduce the 3D tracks. And finally is a software solution to do this. Because if anybody has done anything to do with video footage and 3D tracking, what you end up with is a different software solution for every single experiment you do. Because everything is ever so slightly different and you need to spend hours and hours writing programs. So what we're developing is it's a drag and drop software solution where you can simply say, okay, I need to have a 3D tracking module which drops in. We have the 3D footage and we drop that in, and it's simply linked together as a flowchart of what processes you want to happen, which stops the researcher having to spend hours and hours programming Python or MATLAB or anything else, so that you basically get the data that you want out of the system. without spending that amount of time programming. And at that, I'll leave you with us.
So that's all the kind of work that we're doing that revolves around kind of remote sensing and monitoring. We're just coming on to the other aspect of what we do within MTRU. So we can 3D print some of these 3D models that we've made, and there's some examples over there on the table as well. And this is really motivated by the idea that when you want to put in an artificial structure underwater, you want to try and create some of the complexity, some of the natural complexity that you're trying to emulate that isn't there in the first place. And this is an example of a sandstone printed artificial reef that's probably about 1.5 meters cubed, say. So it's a fairly sizable structure, but it's made using what's called a subtractive printing process. So it's basically layers of sand glued together in a big cube, and then the waste material is washed away and thrown away. And what we wanted to try and do was emulate complex structures, natural complex structures, using 3D printing. And so we took a 3D model of this coral skeleton here, and then 3D printed it. And we can actually print to a resolution of about 0.2 millimeters. So it's highly accurate. The problem that we have is actually being able to support the structures that are so complex. So this is our Delta printer that we have in the department. We have a couple of different types of printers, but this is one of the ones that it swings from the top rather than builds from the bottom. And you can see there on the left that these little spikes are coming up, and these are supports to support the overhanging coral structures that have been built. So they get snapped off and cleaned away, but effectively it's what's called an additive 3D printing process. Now, of course, we don't really want to be putting plastic on the reef with these structures, so we have also been experimenting printing 3D models in clay. And if you thought printing corals in plastic was hard enough, printing them in clay is virtually impossible. Hey, that's research, right? We like difficult things. So it's a very difficult thing to do. The good news is that it's a natural material, and corals prefer to grow on these kind of materials. And it has a low CO2 emission in comparison to things like concrete. However, you do have to kiln it to make it hard, and that's obviously has an impact on its ability to do it out in the field. Really, it's very difficult to actually get these things printed. But that's another one of the things that we're looking at. So we can 3D print these things, and they're very engaging for the public to actually sort of physically get their hands on bits of coral. The other thing we can do is actually create immersive environments and allow people to walk around these kind of 3D models that we've made. And that's where I'd like to introduce Rachel and her work. It's called Crab Fight. This one.
So I'm here to talk to you about crab fights. So yeah, yes, we've built there, change the promotion. So anyway, crab fights is taking a game purpose. So if you're familiar with the concept, basically it's an approach to research where if you have like a complicated thing that you're trying to get data about, you basically just turn it into a game, and that game which can allow the data to be used and play it, and then basically get people to play it. So you generate the data you need, and then profits. So crab fights itself. So it's the game of purpose. And brief model is basically to assess how environment complexity affects like the marine world's life. And it does this through the medium of online multiplayer PvP combat. Yes, seriously. So players has the local wildlife, the Flemish Toolkit, these marine conservation zone and they need to survive. Uses and because of the photographic stuff, it's in real locations. Yeah, it's first person perspective thing. So yes, yeah, you become crab. Specifically, they said that's predators. You gets straight quickly like a crab, and then forces sustenance. Brutally merged by other crabs to assert dominance, just like in real life. So, yeah, basically, it's RIOS, except if you that's except as person into yeah, it's just people getting stronger, then these it also to sustain itself. So and then try to what's on side. And yeah, it's and then games also most sports and it's built with Unity uses the method objects which the multiplayer. Anyway, more important bits. It's how to actually fulfill a purpose. Basically, when you're anything that happens it's all as in a blog file with this sort of structure. So to give you the gist. Basically, every single school club has its own crab-like thing, and then basically whenever that player does anything, the current state of the club, as well as the time since the game started, is logged, and same with the food things as well. So whenever anything of note happens to them, that's logged. So yeah. So if you do anything in the game, so everything has to be logged. Literally, so let's go on. Yeah. That's anyway. If they point is that once there's updates, they say the analysts see rich moments and for that cycle times etc. So it's try something to protect the basic requirements and also it records like locations, the location of time, and because records where everyone was at all times, and then they just show that's protects them arena and so good. And also there's the obvious PR split PR things universities, yeah. Anyway, so if it wants to check out, it's available great. It's that. So I'm just getting a bit. Yeah. That's the sense. Yeah.
Just look back there. So we'll actually have a go at trying to play crab fight together in the room. You can fit 10 players playing at once. So we've got, I think, 4 or 5 laptops here. You can download it. It works on a mobile phone as well, not iOS, but Android. So feel free to download as we just finish off the slides, and we'll have a go at actually trying to play it live. I can't imagine anything going wrong. Just a brief mention then about some other aspects of biomonitoring. So this is actually part of my PhD work way back when Facebook was popular, and Cambridge Analytica wasn't even a thing. The way divers and sort of people who are interested in marine life were connecting through Facebook groups, it was very common to use them as a way of identifying things. And this is what you see in this process here. is you go out, take some pictures of animals on the reef. You take a picture of something down on the bottom left there. What's that? Anyone know? Yeah, you only know that because of Facebook, right? Yeah, right. Yeah. So yeah, it's a warty sea slug. So this I did did this as an example in another presentation, but I posted that up during the presentation at the beginning. With 8 minutes, I had scientific name and a common name, and also so often you get some interesting stuff about what they eat and their interactions, and all that's coming from. I don't know any of these people. I've never met them before, but they just happen to be in the same Facebook group as me. People who like sea slugs. I won't go into that too much. But it's an interesting problem from a language processing point of view. So back in the day, it was deemed okay to access social media conversations and analyze them. Perhaps not so much now. But you could access lots of interesting things. So you could get the image itself, the locations it's taken from, common names, the real names, interactions, all this kind of stuff. And being able to make sense of that, and also being able to make sense of people agreeing, disagreeing with each other. It was a really interesting problem in terms of collective intelligence, and I did a lot of work on it. Cut a long story short, actually these groups, those specialist groups looking at marine life, were doing a much better job of identifying very difficult images than other ways of engaging crowds. So Crowdflower is like a collective intelligence platform where you can upload images and get people to try and annotate them. But it seems on Facebook, people are doing a very, very expert job trying to do this. And the kind of things you can use that for, once you've sort of cleaned up that data, is to build up a library of images so that you can very quickly look at the morphology with something like this, which is Genolus cristatus, UK, quite a nice UK sea slug. And immediately just look, these are all Facebook images that came out of the system. You can see the serrata, which is the long pointy bits, and the blue tips on the end, and you can see the peduncle in the middle of the rhinophores and all that kind of stuff. Just using images from Facebook to get a really good idea of species morphology. So those are the kind of things that we were doing using natural language processing to do biomonitoring and social media. So those are the kind of things that we've been working on. That's the kind of research we've been working on within CSEE, and obviously collaborating with different partners and people within life sciences as well. I'll reiterate that bit at the end. We're always looking for new collaborations and ways to innovate. So if you have any ideas, if anything like that has piqued your interest, then do let me know. I'll just mention a couple of other things. So a few years ago, I think around about the start of the pandemic, I was interested in doing research in the UK. And to do that, we needed to become an HSE registered contractor at the University of Essex. So we've done, cut a long story short, we did a load of paperwork to make that happen. So we can now go out and organise our own research trips, diving research trips in the UK. And we work under what's called accepted codes of practice, or ACOPs. And there's different types of ACOPs for different types of work, but we would typically work under the scientific one. And because of that, that allows us to not only organise our own dive trips, our research trips in the UK, but we can also now collaborate with other projects. So we did some work in the summer with Michelle looking at eDNA seahorses down in Studland Bay, which is great. And we can also join Natural England and Natural Resources Wales and also other partners for the different types of marine surveying projects that they do, because we're sufficiently qualified and have the right equipment to join in. And that's actually a very beneficial position to be in, because there's lots of groups that are quite small, and particularly with the whole pandemic, people testing positive and everything, we were asked to join a lot of surveys because people were dropping out and they needed the cover. So that's a really good benefit. If you're interested in professional scientific diving, then this is the place to start with. I'm really pleased to say that over the summer, both computer science and life sciences contributed to investment in diver equipment, including regulators, BCDs, that kind of stuff, but more importantly, the underwater communications. And I've got the sets of underwater comms over there if you want to have a look at it. But this is effectively what we have. So we have these full face masks. They're commonly used if you're doing HSE-based research in the UK, but we actually want to try and adapt this for underwater. I hate to say lecturing, because that sounds like you're just going to be like 2 hours of talking through this thing, which is not what we're going to do. But we'll be using them on the biology field trips. in the tropics, because I think that would really be able to get information across to the students whilst they're diving. So that's a really exciting project for next year. We also have some other interesting bits of kit that we're very happy to use on different types of projects. So we have the DTG3 Deep Tracker ROV, and that's over there in the corner as well, if you want to have a look. That can go down to 200 meters. So we hope to use that for some deep surveying next year. And we can outfit that with cameras all over that to do photogrammetry and rapid surveying. We've also got cameras, GoPros, we've got a inflatable boat, a diver propulsion vehicle, all sorts of things. And it's all basically trying to build up a sort of equipment that will support teaching and research in these kind of fields. We do collaborate from a teaching perspective as well. So I've had joint master's projects with Boyd, and that's gone really well this year. And we've also proposed a number of life sciences themed projects. And they can be either joint or paired. So by joint, I mean they can be jointly supervised. One student works on one project, or they can be paired. So we have two students. One student is working on a computer science engineering aspect. One student is working on a life science type of research question. And they work together, but they are separate projects. So we can do that for undergrads, masters, and PhD level, just depending on who we've got and what we're interested in. As I mentioned before, we've had internships. So we've had the coral biology students working with us doing image analysis, but also building frames and doing other kinds of scans and that kind of stuff. This weird image that you see is what we did on Monday. I gave a talk on photogrammetry to the BS 333 students. And we very quickly passed a GoPro around the room, and we built a 3D model of the classroom with the students in there. So it's a very rapid way of capturing data. So just to wrap up, this is really all about trying to improve the toolkit that's available to people that are interested in marine conservation and marine research. There's no silver bullets here. This is all kind of stuff that's work in progress. And there are sort of bounds of error that you have to accept within it. But it does allow you to speed things up. It allows you to scale up our response to trying to address this issue of the reduction in diversity and complexity on reefs. And hopefully there are ways that we can use this technology so that we're not just watching the decline of the reefs, we can actually address it, try and reverse it as well. So if you'd like to have a go at Crab Fight, that's the web address to go to. You need to download it and start it up. You'll get some kind of sinister looking alert that says, are you sure you want to run this exe file? So just ignore all that and run it anyway, or don't, as the case may be. But yeah, you'll need to ignore that security warning. And what we'll do, I'll take some questions now, if there are any. And whilst I do that, Rachel's going to set up the game. We're going to attempt to play it live in the studio. Thanks to Mark, thanks to Rachel, and also thanks to Jess, who got here fighting through the traffic. Would anyone like to play the game? Okay. Thanks. Usually the kind of metrics that we just use the mesh, because you don't really need the texture for anything. You just need the kind of the shape of the object. So like rugosity or something like vector dispersion, those kind of things. You only use the mesh. But if you were looking at things like coral bleaching, for example, you would need the color. And yeah, you could use it in any way you want. You would obviously need to develop programming, but you could, for example, look at bleaching on the tips of corals, for example, which is something that to do in situ would be an active nightmare. But to do it programmatically, you could make something to do that. Yes. I think so, yeah, and also repeatable, yeah, as well. So in terms of the data, you can go back to that and keep analyzing it. The type of photogrammetry we do uses kind of GoPros on a low setting, so it's not very detailed in comparison to sort of using like a digital SLR. So some projects, so like with NOAA and CoralNet and those kind of projects, use very high resolution imagery, produce kind of polyp level detail, which is amazing, absolutely amazing to see. But we kind of went down the road of using cheaper cameras and having less resolution to make it, and it's also more of an accessible technology. Yeah. Can I say maybe? Yeah, in principle, yeah. Yeah.
So, you want to sort of keep the question back to people on Zoom.
Sure,
So I
sure.
was going to ask you about it's like to get fish weight in the field, right? And so some of the stuff we're trying in California is taking an image of the salmon on like a little, with a little kind of gridded set, and then using the automated image analysis to try and get detail of it. Have you played anything like that? Please really get that.
Using photogrammetry.
Or just like more connected, so like finding or using AI to find anchor points. You'll like click on the snout and the tail.
Yeah. Okay, so just repeating for the people on Zoom. So Anna was saying, can we use any of these technologies to measure weight and size? Yeah, so that's not something we've done, but we have worked with Eastern IPCA, and they're very interested in that from the perspective of measuring crabs and lobsters, particularly the carapace size, because that is they use that for landing measurements. And they, I think it's Newcastle that developed a bit of technology where you basically plonk the lobster. There's a camera facing downwards, and it does a little beep. Goes, yeah, that's okay, or it doesn't beep, and then it has to be chucked back. So there are technologies to do that live and in the field. In terms of doing it for sort of marine monitoring, sort of like a post hoc analysis, I think if you've got the images, you could build something to do that. It's just about identifying the right areas of fish.
Basically, we put on 20 seconds, and then we go and do it for still needs to be validated.
Yeah. Okay. I'd love to still do that because they're all talking about the quality of the habitat, but then because the length of it is not that telling the fact.
Yeah.
Yeah, but that's where if you want to this habitat better, that.
Yeah, well, like every single fish, in fact.
No, my point is like for that one species, the one that they're back there.
Yeah, that probably would be they basically land.
Yeah, exactly. Sorry, future project.
Yeah, future project. Yeah, future project. Any questions on Zoom? Is anyone still?
On my one, sorry.
Yeah, there's no questions on Zoom.
Any questions on Zoom?
Yes.
Philippe.
Hi. Thanks a lot for actually I can for what it's worth. Thanks a lot for a nice talk. One of the things with AI, certainly in terms of microscopy, is always knowing the ground truth. And so in your case, you do that with annotation from students. But do you think there will be, will there be a way to make AI decisions more obvious so that you can actually follow them back and see where the program went wrong in terms of determining a structure or an organism. So have more, will AI ever be auditable?
Yeah, good question. Yeah, Philip, I'm gonna have to repeat that because it's just come out of my laptop, so these didn't hear it. So
Okay.
Philip saying, is the AI processes can it be auditable? So can we detect where it's going wrong within these kind of image identification processes? And there is a big research theme or funding floating around for explainable AI in lots of settings, particularly in healthcare. So if automatically you've been diagnosed with something, you're going to be very interested in knowing how that diagnosis came about, whether there's humans involved or whether it was automatically generated. And the same applies for all AI technology, really. I think a lot of people treat deep learning approaches as a bit of a black box. You type the magic words, stuff the numbers in, numbers come back out the other end, and people don't really understand the mechanism behind it. And that's really where explainable AI comes in, is to try and help people understand what's going on with those processes. You also mentioned about the the gold standards, the labeling data. And there are ways people are trying to do training with very little labeled data, but yeah, with mixed results, I would say.
Okay, thanks.
Any other questions, or should we have a go on the game? Let's play. Okay. So I think someone can play on this laptop. Someone can play if anyone's logged in. Is anyone on Zoom logged in? Give us a shout if you have. Okay. Yes, what's your name? Alice. Okay. Oh yeah. Oh, are you coming in here now? Okay. Okay. How are you doing? Yeah, good. Good. I think what we might do, we might transfer ourselves to another location.
Okay, you were planning to do it, still doing it.
Really, we
Like
do
the
it.
sun were getting up, then I thought I can go.
Yeah, yeah. No, we can do a
getting
little demo, but I think it might take a moment or two to begin. So I think.
Okay.
So thanks everyone for joining on Zoom, we're going to try and go to a different location and have a go at crowd fight. So if you're on Zoom, join through the game, and we'll see you there in a second. Anna, we could go to a different lab. Yeah. Okay.
Thank you. That's brilliant. If you throw up cool stuff. Okay.